Here we have our normal coding sequence of DNA and 4 mutant coding sequences with single nucleic acid point mutations. Here's the DNA sequence with no mutation. The first step in translating this sequence is translating it to the mRNA level, which is the same sequence as the coding strand, just T's are replaced with U's. To start the reading frame, we look in the 5' to 3' direction to find the first AUG, which is right here at the beginning. Next, we'll group the nucleic acids in groups of 3, and using the DNA codon table, we can translate the sequence into the following. Let's compare our original coding strand with point mutation A. We see that the mutation has changed the base from C to G. This is a transversion in which the base changes from a pyrimidine to a purine. Looking closely, we see that GCU was changed to GGU. GCU translates to alanine, whereas GGU translates to glycine. This change in the amino acid coded is known as a missense. The single base change changes the amino acid and can also change the function of the protein as a whole. Next, let's compare point mutation B to our original coding strand. In these two strands, we find the mutation here where base G has changed to base A. This mutation is known as a transition, in which the bases change from purine to purine. If we look at UGG and UGA, where the mutation occurred, we can see that UGG translates to tryptophan, and UGA translates to a stop codon. This abrupt stop is known as a nonsense mutation. We have changed a codon, tryptophan in this case, to a stop codon. Moving on to point mutation C, we find the mutation with this T changing to a base A nucleic acid. This is another transversion mutation where we see the pyrimidine T change to purine A. Looking at the mRNA level, we have GCU from the original strand that codes for alanine, and GCA from point mutation C that also codes for alanine. This is known as a silent mutation, where the DNA was changed but was coded for the same amino acid. Lastly, looking for the mutation between point mutation D and the original coding strand, we find it here where T changes to C. This mutation from pyrimidine T to pyrimidine C is another example of a transition mutation. In the original strand, we have UGA that translates to a stop codon, where in strand D, we have CGA that translates to arginine. Changing this stop codon to an amino acid is known as a read-through, where instead of stopping, the strand will continue to translate into a longer protein.